If you remember, in the introductory chapter to biology, we talk about all living organisms need to acquire energy. And we didn't spend a lot of time talking about it. The main reason is because there are 3 videos coming on that topic. This is what we're going to be talking today in general, and then we're going to have another couple more of videos how energy, sun energy is converted into chemical energy and how chemical energy is converted into the form of energy that cells can use. But those are going to be 2 different videos. Today is just an introduction. Okay? But we say cells need energy, and organisms obviously, to stay alive. They need to grow. There's going to be damage to be repaired. They do work. I mean, even if you look at the cell level, they have to be chasing infectious bacteria or digesting food or responding to stimuli, whatever. Cells are constantly working. And without energy, they cannot build those macromolecules I was talking about in the biochemistry chapter. They can move materials in and out of the cell. Remember, active transport requires energy. They cannot get rid of waste. So, as an example, you have here a hummingbird. If you've ever seen a hummingbird, how it hovers, how fast it moves the wings, how fast it flies when it goes from one place to another, it requires a lot of energy. So the hummingbird is going to eat what you put in the feeder or directly from a real flower, and it's going to digest that food, and through a series of biochemical reactions, will get energy. We're not going to be talking about actually ingesting and digesting food. We'll cover that in the second semester, okay? But we are going to be talking about once the food is in the body, what do we do with it? So all the chemical reactions in the cell either release or require energy, okay? So if you're using energy, obviously you have to be producing more energy. So those other reactions take place. All of the chemical reactions that take place inside the cell, where they produce or release energy, they are known by the name of metabolism. Now, we said in the introductory chapter that ultimately the energy comes from the sun. Plants use photosynthesis to capture sunlight, and they make chemical energy, so that's, we call them producers. Now, herbivores are animals that eat plants, and they get energy from the plants. Carnivores eat the herbivores, so both of them are called consumers. And eventually they all die and they decompose due to the decomposers. Okay, so we're going to be studying all of these different levels of energy use and energy formation. A metabolic pathway is that series of chemical reactions that takes place with a molecule. It modifies it. It's not just a one big complicated reaction, but usually a series goes through several steps until you get to the final product. When you generate energy, that series of reactions is called a catabolic pathway. And when you need energy, when they require energy because they're synthesizing bigger molecules, that's called an anabolic pathway. And I don't know of any mnemonic to remember which one is which. I'm sorry. No, we are not going to cover thermodynamics in detail, I promise you, okay? I have probably 4 chapters in my chemistry videos. No. I just want to mention thermodynamics is the study of energy and energy transfers. And of course, we're going to be talking about energy, so there's a couple of things I do want to introduce in here. We say energy is defined as the ability to do work, but it appears in many different forms. You can have kinetic energy, that's the energy of motion. When your car is going down the road, it's moving, that's kinetic energy. Potential energy is energy that is in an object because of its composition, maybe because of its position. For instance, you buy gas for your car. Gasoline has potential energy. If you just put gas in the car is not going to move automatically. You need to burn that gas. That potential energy needs to be released, and then it becomes kinetic energy when your car starts moving. There's heat. We talk about how in all of the transfers, heat is usually lost. Think about your car. When you make a long trip, what happens to the engine? It's hot, right? And we're not going to get into free energy because that's more of a, excuse me, chemical term. Don't worry about it for now. But the first law of thermodynamics says that the amount of energy in the universe is constant. Energy is not created, it's not destroyed, but it is transformed. Like the example I gave you, your car gasoline has potential energy. When the car is moving, that potential energy becomes kinetic energy and heat energy. It's the same amount of energy, just transformed from one form to another. We're not going to worry about this definition because I'm not going to worry about free energy, but reactions that release energy are called exergonic. And some people call them exothermic too. Okay? Other reactions absorb energy. Okay? So they're called endergonic or endothermic. Biologists tend to like exergonic and endergonic more. Chemists prefer exothermic and endothermic. Same idea. And you may want to use whatever your instructor uses. Here you have examples of energy transfer. An ice cream cone, food, has potential energy in the bonds of the molecules that make the food. But then you eat it, and then you're up to go for a walk, ride a bicycle, whatever, it becomes kinetic energy. Sunlight is light energy, but when the plants do photosynthesis, they make chemical energy. It's a form of potential energy in the bonds of the molecules that you made. Another example, still water, like in the dam, has potential energy. There's energy in there, but it needs to be used. It needs to be transformed to something. If the water stays there, it's still potential. But if you open the gates and energy flows, or like in a waterfall, then all of a sudden you have kinetic energy. And I'm going to go a step ahead. If you have a gate in here that goes to a turbine, yes, the water goes through a turbine, has kinetic energy, but what does a turbine produce? Electricity. So you found another form of energy, electric energy. But like I said, we don't create, we don't destroy energy, we just transform it from one form to another, depending on the needs. I want to introduce enzymes because we are talking about chemical reactions. Enzymes are proteins. We did see that when we studied macromolecules. They're catalysts. That means two things. One, they speed up chemical reactions, and two, they are not used. So the enzymes remain there to keep on catalyzing. Just to give you an example, years ago I was teaching at the high school, and I brought two beakers, and I put them on my desk. One has hydrochloric acid, because that's the acid in the stomach. The other one has hydrochloric acid and the enzyme that you find in the stomach. And I had two little pieces of a steak that was from the night before. It was a very good excuse. And I put one on each beaker and I stirred them. The next morning, I did the same thing. The piece of steak that had the enzyme, but the second day when you stir, it basically dissolved, it disappeared. The one that only had the acid, we kept doing it every day, except for the weekends, of course. It took almost 3 weeks. So that gives you an idea how the enzyme speeds up the reaction of breaking down a protein. To understand how enzymes work, you need to understand energy of activation. That's the energy that you have to add to a reaction to get it started. Go back to your car. You have your tank full, you go there in the morning, and the car does not start. What's the problem? Well, maybe you're very unlucky and your starter broke. Most likely, your battery is dead. And the car won't start because you have to give energy to start the car running. Now, once the car starts running, the battery doesn't do anything, unless you have an electric car, obviously. Your car runs on the gas, but you have to add that electrical energy to begin with. So that would be an example of energy for activation. You have a chemical reaction, you add energy of activation. What the enzymes do is they lower the energy of activation, so that makes it easier for the reaction to get started, so they increase the reaction rate. It's kind of like, again, with the example of the car, what if instead of a car battery, you could start your car with a couple of AAAs? Have you ever had to replace the battery in your car? It's expensive, isn't it? But if your car could use AAAs, you'd be better off, because it doesn't happen that way. But that's what an enzyme does. Okay? Now, enzymes are going to have a section of biochemistry completely devoted to them, so they kind of have the right to change a few names. So the reactants in the reaction characterized by enzymes, they get a different name. We call them substrates. The location in the enzyme where the reaction takes place is called the active site, and the active site actually holds the reactants in place for the reaction. And the other thing that we need to keep in mind is that we said enzymes are proteins. So they can lose their shape because of the environment, where you talk about temperature or pH or number of factors. If they denature, remember that means they lose their shape. And a protein that loses its shape loses its function. So enzymes that are denatured don't work. This is the latest model of how the enzyme works. We used to think that the enzyme and the substrate were like a key in a lock, because enzymes are very, very specific. So if you put the wrong key in a lock, you cannot open whatever it is. We know now that it's not very accurate. We have the induced fit model saying that actually the enzyme, the active site of the enzyme and the substrate are very, very similar, but not completely identical. When the enzyme binds the substrate, now the substrate changes a little bit, or the enzyme changes over a little bit, where now it fits perfect. But it didn't have to fit perfectly at the beginning. Now, once they fit perfect, then the reaction takes place and the products are released. We're going to talk about other molecules that sometimes bind to an active site. The only way you can explain this by saying the active site is going to bind molecules that are very, very, very similar. If it was a key and lock, there'd be no way for anything but the substrate to bind to the active site. But we know that's not the case. But it has to be something that's very similar to the active site, where the active site can adjust. So since enzymes are proteins, they're going to be influenced by environmental factors, and that allows the cell to control enzyme activity. One factor could be the substrate concentration. As the enzyme activity increases, when the substrate concentration increases. Cannot make sense if you have, let's say, 1,000 molecules of enzyme. If you have only one molecule of substrate, you can only change one molecule. But if you have 10, you can change 10. If you have 100, until you reach the limit of how much enzyme you have, the more substrate you have, the more product you get. Makes sense, right? Temperature. Enzyme activity increases with temperature. Proteins don't like cold, okay? So you can see in there that at low temperature, activity is very slow. As the temperature increases, enzyme activity increases, but at a certain point, it gets too hot. The enzyme is denatured, and you can see the drastic drop in enzyme activity. Most enzymes have an optimal temperature, that is the temperature of the organism in which they live. The pH, the acidity, is going to influence enzyme activity. But the optimal pH, the one at which the enzyme is the most active, varies depending on the location. For instance, pepsin is found in the stomach. That's the enzyme that I was talking about that is present in digestion and gastric juices. So it has a pH, optimum pH of around 2, because that's the pH in the stomach. It needs to be able to work in the stomach. But if the enzyme is located like trypsin in the small intestine is not going to be acidic. So it has an optimum pH Yeah, but between 7 and 8. Every enzyme has an optimum pH for the location in which it's working. So how does the body regulate enzymes? We are going to be looking at the four main forms of enzyme regulation. Once again, I'm just going to read the list and then we are going to explain each one. But you have competitive inhibition, non-competitive, also known as allosteric inhibition, allosteric activation, and feedback inhibition. So competitive inhibition. Some inhibitors can bind to the active site. They don't have to look exactly like the substrate, but they're close enough, like in this case, you can see it binds to the active site. So now the substrate cannot bind because there's something else in the active site. Okay? So you call that competitive inhibition. And usually, not always, but usually this is reversible, meaning if you have a bunch of substrate, it can rinse away the inhibitor. Okay? But sometimes it's irreversible. You have a competitive inhibitor that binds and holds on for dear life. Non-competitive means that there's no competition between the inhibitor and the active site. Instead, the inhibitor binds to another site, that's not the active site, we call it the allosteric site. You see, the inhibitor is binding, the active site is the round thing, is binding to the allosteric site. When it binds, it causes a conformational change in shape in the active site. So now the active site cannot bind the substrate. You see this substrate here, it would fit perfect with the active site. But once the active site has changed shape, it cannot bind the substrate. So it's allosteric because it does not bind to the active site, and it's inhibition because again it prevents the substrate from binding to the active site. But you have also allosteric activation. This is kind of unusual, but it happens sometimes. You have an active site that you can tell in there it cannot bind the substrate. You have the substrate that's around it, it's never going to fit in there. But an activator binds to the allosteric site and causes again a conformational change. Now the active site has the right shape to bind to the substrate. So it's allosteric, again because it binds to the allosteric site, activation because it turns on the engine. Now the enzyme can work. Finally, feedback inhibition. We talk about metabolic pathways as a series of reactions, and usually you have multiple enzymes. For every reaction, you're going to have a different enzyme. Sometimes you have more than one enzyme that can be controlled, but usually it is the first enzyme that controls everything, because if you control the first step, obviously that controls everything else. So many times the end product gives feedback, that's what is called feedback inhibition, to the first enzyme. It works as an allosteric inhibitor. So it stops the first enzyme from working. Why do you want to do that? Well, you're trying to make something that your body needs, but at a certain point you've made enough. You don't need any more. So whenever there's enough end product, it stops so the cell does not waste materials and energy making more of a product that you already have. The last thing I want to talk is about ATP, because this is the molecule that the cell uses for energy. Okay? This is a nucleotide, you should recognize it. You have a nitrogenous base, that's adenine. You have a sugar, ribose, and you have 3 phosphates. Okay? That's what ATP means. Adenosine, that means the adenine and the ribose, triphosphate. Now, the bonds between phosphates are very high energy bonds. Okay, and this is what the cell is going to be using. So you have your ATP. If it loses one phosphate, now you have ADP, adenosine diphosphate, because there's only 2 phosphates. And the energy is released for the cell to use. Now the cell has excess energy and it wants to store it. So ADP can pick up a phosphate, but it takes energy to do that and becomes ATP. So ATP functions like a rechargeable battery. When you need energy, you take it out of the ATP. Whenever you have excess energy, you recharge the battery. Isn't that a neat system? So I know this wasn't very complicated, but once again, if you want to work with me one-on-one, if you want any of the charts I have made to summarize different reactions, if you want to study for an exam, there's a link to my Washington profile in the video description. All you have to do is make an appointment and we'll get together.